Okay, so the last piece of what we're going to talk about the innate immune system is something called complement. And essentially, this is a complementary series of actions for the immune system. And basically, it consists of a whole series of proteins that are found in the blood that work in concert to destroy bacteria and certain viruses and parasites as well. It's a cascade reaction. In other words, one thing happens and the next thing and the next thing. It's a whole specific sequence of events that happen. And the end result is that it will pop the bacterial cell open or whatever. So the end result of all of these actions, one protein landing and then the next and the next, is that it makes a hole in the cell membrane. Okay, so again, this is referred to as a cascade. So one thing happens, then the next that falls over into the next and the next. Okay, so there are a whole bunch of complement proteins, and they are all numbered, and in some cases you have a number followed by a letter. Okay, so the first thing that happens in the cascade is the C3 protein lands on the pathogen, and it's broken into two subpieces, C3A and C3B. Okay, so then C3B cleaves another protein called C5 into two pieces. The C5B fragment is now free to form a complex with C6, 7, and 8. And all of those pieces together are referred to as the membrane attack complex, or MAC or MAC. The membrane attack positions itself on that cell membrane and forms a pore, and the cell membrane then kind of loses its structure and all of the internal parts of the cell leak out into the cytoplasm. Okay, and basically it pops the cell, it lyses the cell. That's the end result of the cascade, the complement cascade. Okay, so there are actually two different types of cascades. One is referred to as the classical complement pathway, okay, and it's initiated by binding to the antibodies that are already attached to the microbe. And then there's the alternative complement pathway, which doesn't need an antibody to get started, okay, and it's just simply initiated by the presence of foreign cell antigens. Okay, and it's quicker than the classical complement pathway. Okay. So here is an example of that cascade in a picture form. Okay, so the initiation is C3 being split into two. C3 combines with a piece of C5, which is cleaved as well. C5b attaches to the bacterial cell, and then several other proteins land, and it makes that that MAC, that membrane attack complex, which lyses the cell. Okay. So the last thing we're going to talk about in terms of the innate immune system are these antimicrobial peptides. And those are really, really short sequences of amino acids, so maybe 12 to 50 amino acids. And an example of that is defensin, and that is a sort of the prototypical antimicrobial peptide. And defensin is able to essentially insert itself into a bacterial membrane and thereby disrupt the structure of that membrane. So if you think about it like, pounding a nail into the membrane, and, that makes it lose its structural integrity. Again, it creates a pore in the membrane, and if enough of those happen, then the cell will lyse. It also has an effect on other actions of non-specific and specific immunity, okay? So these antimicrobial peptides are kind of a natural thing, and researchers are now looking at those antimicrobial peptides to see if we can actually turn those peptides into a drug. and insert those into the body exogenously, and sort of beef up the immune system to be able to attack certain bacteria, particularly ones that have become antibiotic resistant. It's pretty hard for bacteria to resist getting a hole poked in its cell membrane. That pretty much will kill it no matter what, and there isn't a whole lot of resistance that it can develop to that. So here's an example of what that would look like, what a defensin would look like. So there is a part of the peptide that is hydrophilic, so it's perfectly happy out in the aqueous environment. And then there's a piece of it that's hydrophobic. So the hydrophobic piece will actually insert itself into the cell membrane through those phosphate groups at the top and down into the lipid layer of the cell membrane. Okay, and again, you can sort of see how it's kind of pushing those phospholipids out of the way. Well, if you get enough of those, you're going to get everything completely through the membrane, and then it will lose its integrity and all of the contents will leak out, and that's the end of the bacterial cell. Okay, so here we're going to talk about, we're going to go back to phagocytosis, and we're going to talk about the order of phagocytosis. So take a look at this. It will help if you sort of think about what is the very first thing that's going to happen in the process of phagocytosis. Okay, so let's go down the numbers. Engulfment is when it actually wraps around the bacteria and pulls it in. The killing and destruction of the cells, that's pretty self-explanatory. Phagosome formation, okay, so once you have the bacteria encased in the cell membrane, that is the phagosome, okay. Adhesion of bacteria, so when does that happen? Release of residual debris. Chemotaxis by the phagocyte. Okay, remember what chemotaxis is? That's a chemical is released and that causes cells to appear at a particular place. So something has detected an infection has happened, it sends out a chemical and all the other cells come running. And the phagolysosome formation. Okay, so this is adding the lysosome piece to the phagosome. Okay. So just to give you a hint, number 6 is the first thing. So somebody recognizes that there's an infection happening, it sends out those cytokines, and the macrophages come running. Okay, so that's the first thing. The macrophage has to show up where the bacteria are before phagocytosis can happen. Okay, so we're down to B and D. So what would be the next thing, 4 or 3? Adhesion of the bacteria or phagosome formation? If you think about what the phagosome is, that is something after the bacteria has been engulfed and it's been wrapped in the membrane as part of the whole phagocytosis piece. The 2 pseudopods form, wraps around the bacteria, the piece of the membrane detaches from the phagocyte. So that would be, the adhesion of the bacteria would be the next thing. So the macrophage comes running and then grabs onto the bacteria. And then, so the answer obviously is going to be D. So the next step is engulfment. The next step after that is the phagosome formation. So after it engulfs it, it forms that bit of membrane around the bacteria. And then the next step is phagolysosome formation. So lysosomes with all of those digestive enzymes then become a part of the phagosome. And that begins the digestion process. The ultimate result of that is the destruction of the bacterial cell. And then the last part of it is release of residual debris, because after the cell is dead, you want to get rid of the excess stuff. And then you want to take those antigens and display them on the outside of the macrophage in order to do the handshake with the specific immune response. Okay, that's going to be the end of the material for Chapter 12. I will see you in Chapter 13.